السلام عليكم طلابنا الأعزاء. اليوم ناخذ موضوع الهيموبويسيس. Haemopoesis. Mature cells, blood cells, have relatively short lifespan and must be continuously replaced with the stem cell progeny produced in hemopoietic organs. Haemopoiesis means formation of blood cells. Heme mean blood, poietic mean making or formation. In the earliest phase of human embryogenesis, blood cells arrive from yolk sac mesoderm, while in the second trimester, hemopoiesis called hematopoiesis occur primarily in the developing liver, with the spleen also playing a role. In the second trimester, mean occur in liver and spleen, while in the earliest phase occur in the yolk sac. In the third trimester, bone marrow increasingly became the major hemopoietic organ. After birth and on into childhood, erythrocytes, granulocytes mean neutrophil, eosinophil, and basophil, and monocyte and platelet are derived from stem cells located in the bone marrow. The origin and maturation of these cells, termed erythropoiesis, according to the formation of the process of erythrocyte formation, granulopoiesis, formation of granulocytes, monopoiesis, monocytopoiesis, according to the formation of monocytes, thrombocytopoiesis, according to the formation of platelets. Development of major type of lymphocyte is lymphopoiesis, occur in the bone marrow and in the lymphoid organ to which precursor cell migrate. Before reaching maturity and being released into circulation, blood cells go through specific stages, differentiation and maturation. Because these processes are continuous, cells with characteristics that lie between various stages are frequently encountered in smears of blood or bone marrow. The main cell in the bone marrow is pluripotent hemopoietic stem cells. This is the main cell that all blood cells differentiated at maturation from this cell. It is believed that all blood cells arise from single cell type اللي هي هذه of stem cell in the bone marrow called pluripotent stem cell because it can produce all blood cell types. The pluripotent stem cell proliferated and formed two major cell lineage, one for lymphocyte اللي هي lymphoid cell and other for all the rest blood cells اللي هي من جزء المyeloid cell. Myeloid cell include granulocytes, monocytes, erythrocytes, and megakaryocytes. Lymphoid cell migrate from bone marrow to thymus or to the lymph node or spleen and another lymphoid structure where they proliferated and differentiated. This is the pluripotent hemopoietic cell, the one stem cell that all cells originated and differentiated. Divided into two cell lineage, myeloid stem cell and lymphoid stem cell. Lymphoid stem cell differentiated to lymphocyte, while myeloid stem cell اللي هي progenitor cell give three progenitor cell erythropoiesis for colony forming unit for erythropoiesis, and colony forming unit 2 thrombopoiesis, and colony forming unit for granulocytes and monocytes. All this progenitor cell give rise to the precursor mean blast, here called proerythroblast, and in platelets called megakaryoblast, and in the granulocytes called myeloblast, and in monocyte line called monoblast. This differentiated to form erythrocytes, platelets, granulocytes, and monocyte from myeloid lineage or myeloid stem cells. Progenitor and precursor. The pluripotent stem cell give rise to daughter cell that restricted called progenitor or colony forming unit for erythrocyte, thrombocyte, granulocyte, monocyte, and this is from myeloid, while the lymphoid lineage for lymphocyte. We have two types of bone marrow, red bone marrow and yellow bone marrow. Red bone marrow, whose color is produced by the abundance of blood and haemopoietic cells, while the yellow bone marrow filled with adipocytes and essential excluded haemopoietic cell. Under normal condition, the production of blood cell by bone marrow is adjusted to the body need. إن يحتاج الجسم ينتج البودي cells according to their need. Increase its activity several fold in very short time. Bone marrow and adipocytes are found in the medullary canal and the long bone, and the cavity in the cancellous bone. The newborn bone marrow is red and active in blood cell production, but as the child grows, most of marrow change gradually to yellow variety. Under certain conditions, such as severe bleeding or hypoxia, yellow marrow revert to red. This is the bone marrow. This is the trabeculae that ejected or penetrate the bone marrow, trabeculae from the bony material, compact bone. This is the bone marrow cavity that contains the bone marrow, which is consists of haemopoietic tissue and adipose tissue. All the blood cell maturation and differentiation, as we say, occur in the bone marrow. The first differentiation is for erythrocyte involve hemoglobin synthesis, formation of a small enucleated biconcave corpuscle. The cell and nuclear volume decreased. Big, more small, more to the smallest cell. The nuclei disappear. Nuclei become to disappear. The chromatin became more condensed. Finally, the nucleus, the cytoplasm is basophilic. The basophilia decreased. The basophilia according to the presence of polyribosome. When the hemoglobin amount became to form the here in this stage, the basophilia disappear. Here the basophilia and acidophilia in a balance, mean the hemoglobin and ribosomes found here in a balanced amount. Mitochondria and other organelles gradually disappear. The reticulocytes, the nucleus ejected and the reticulocyte is formed into blood take approximately 3 to 7 days to reach the erythrocyte with differentiated in the blood vessel the glycoprotein erythropoietin, which is essential for production of erythrocyte. This is the blood cell maturation and blood smear. This is the biggest and the most recognizable cell in erythrocyte maturation. Large cell basophilic cytoplasm have nuclei, lacy chromatin, converted or differentiated to other stage called basophilic erythrocyte with a strongly basophilic than the proerythroblast. More basophilic cytoplasm, condensed nucleus, no nucleolus. The basophilia, as I say, according to the presence of large amount of polyribosomes. The next stage is reduced in volume. This is the polychromatophilic erythroblast. In this stage, the basophilia and acidophilia is in balance. يعني between the polyribosome here become decreased, while the hemoglobin became to increase. In the next stage, the cell and the nuclear volume continue to condense. No basophilia filled with the hemoglobin. Therefore, the cytoplasm is acidic. The latest stage is reticulocyte. Reticulocyte have polyribosome and ejected their nucleus, converted and differentiated to erythrocyte that lose their ribosomes and mature. The nucleus of reticulocyte that ejected phagocytize from macrophage. This is the last erythrocyte matured in last stage in erythrocyte maturation. Now, the maturation of granulocytes mean neutrophil, eosinophil, acidophil. Granulopoiesis involves cytoplasm changes dominant by synthesis of protein for azurophilic granules and specific granules. The first stage is myeloblast. This is a recognizable cell in myeloid series with fine dispersed chromatin and faint nucleoli. The next stage is a promyelocyte blast precursor differentiate to promyelocyte. The Golgi apparatus appear. The azurophilic granules, blue and basophil, blue. And this is azurophilic, mean lysosomes. This azurophilic occur or presence in all granulocytes, either in granulocyte and agranulocytes. Neutrophil, eosinophil, acidophil, monocytes, and lymphocytes, responsible for lysosome enzyme and myeloperoxidase. The more differentiated myelocytes, the specific granules appear. Here pink because the cell is neutrophil, while became coarse red granules and acidophil, and coarse purple or blue in basophil. This stage increase in number in specific granules. When the myelocyte more differentiated metamyelocyte, the chromatin is condensed, the specific granule dispersed before complete maturation of neutrophil granulocyte pass through the intermediate stage, called the band stage or the band cell, in which nucleus is elongated but not polymorphic, certain differentiated properties. This is the maturation of granulocytes. While the maturation of agranulocytes as a monocyte and lymphocyte, the stages begin with the monoblast progenitor cell. Is a committed progenitor cell that is virtually identified to myeloblast. Monoblast, myeloblast, morphologically identical one to each other. Further differentiated lead to promyelocyte, large cell, basophilic cytoplasm, slightly identified nucleus, chromatin is lacy, nucleolus are evident, promyelocyte divided twice as they develop into monocyte. During differentiation of monocytes contain extensive rough endoplasmic reticulum, Golgi apparatus complex formation, forming lysosomes, which are observed as fine eosinophilic granules at maturity. This is mitochondria, rough endoplasmic reticulum, eosinophilic granules. Monocyte circulates in the blood for several hours, while when migrate to the tissue become several months. When monocyte migrate to tissue, called macrophage, stay there several months. In lymphocyte, circulating lymphocyte originate from the thymus and other organ as spleen, lymph node, and tonsil. All lymphocyte progenitor cell originated in bone. As lymphocyte matured, the chromatin became more compacted. Nuclei become less visible because of the chromatin compacted and dense. The cell decreased in size. First identifiable progenitor of lymphoid cells, the lymphoblast, large cell capable of dividing into 3 times to form polylymphocytes. Polylymphocytes are smaller and have relatively more condensed chromatin, but none of cell surface antigen that mark T or B lymphocytes. يعني non-differentiated. Prolymphocyte is non-differentiated because the differentiated lymphocyte have T or B receptor on their cell surface. We can detect it for this receptor from the technique of immunocytochemistry by using a specific marker are required to make the distinction. Finally, the platelet, small membrane enclosed formed element, thrombocytes, originated by fragmentation from mature megakaryocyte. Megakaryocyte differentiated from megakaryoblast in a process driven by thrombopoietin. This is the platelets. This is the megakaryoblast. 25 to 50 micrometer in diameter, with basophilic cytoplasm, oval or irregular or kidney-shaped nucleus with several nucleoli. Before differentiating, these cells undergo endomitosis with repeated round of DNA replication, not separated by cell division, result in a nucleus. Megakaryocytes are giant cells up to 150 micrometer in diameter, polyploidy nucleus, and large irregular lobulated with coarse chromatin. Their cytoplasm contains numerous organelles, numerous mitochondria, rough endoplasmic reticulum, Golgi apparatus, from which arise the conspicuous specific granules of platelets. Best seen in bone marrow, megakaryocytes also occur in the interstitial fluid of spleen and lung, most often closely associated with vascular sinusoids or capillary. And thank you for listening.